There is a living organism that can double its own weight in protein every 24 hours. Not in a lab simulation, not in theory, in practice. It does this using sunlight, water, carbon dioxide, and almost nothing else. Pound for pound, it produces more usable protein than soy, more than beef, more than any conventional crop grown on American soil. In the 1960s, NASA did not just notice this organism. They studied it. They funded research programs around it. They envisioned it as the backbone of long-duration space nutrition, a biological engine that could keep astronauts alive on missions where resupply from Earth was impossible. That organism is Chlorella, a single-celled green microalga smaller than a red blood cell. And despite decades of government-funded research validating its nutritional density, the FDA has never approved it as a food. It is classified as a dietary supplement. It carries warning labels, and in certain cases, the agency has actively cautioned consumers against various forms of it. One federal agency spent years and millions of dollars studying how to feed people with it. Another federal agency still will not call it food. That gap did not happen by accident, and the reasons behind it reveal more about how the American food system actually works than almost any crop debate in modern agriculture. The story of Chlorella as a serious nutritional candidate does not begin with the space program. It begins in the aftermath of World War II, when the global population was accelerating and food production infrastructure across Europe and Asia had been shattered. Demographers were issuing urgent warnings about imminent famine. Agricultural scientists were racing to identify calorie-dense, fast-growing food sources that could be produced at scale without requiring vast tracts of farmland. In 1948, a research team at the Carnegie Institution of Washington published findings on Chlorella's photosynthetic efficiency and protein yield. The numbers were striking. Chlorella was roughly 60% protein by dry weight. It contained all 9 essential amino acids. It could be cultivated in shallow water tanks, in nearly any climate, using a fraction of the land and fresh water required by grain or livestock. And it grew fast, not seasonally, not in harvest cycles, continuously, day after day, without rest. Within a few years, Chlorella research had spread to institutions in Germany, Japan, Israel, and across the United States. The Rockefeller Foundation funded studies. Stanford Research Institute investigated scale-up potential. By the early 1950s, serious scientists at serious institutions were publishing papers suggesting that microalgae could become a primary protein source for a world that was running out of ways to feed itself. Japan became the first country to move Chlorella from the laboratory into daily life. By the 1960s, Japanese companies were producing it at commercial scale, marketing it as a whole food nutritional supplement with deep institutional backing. Millions of Japanese citizens consumed it daily. It became embedded in a health-conscious consumer culture that treated nutrient density as a household priority, rather than a fringe interest. In Taiwan, similar production facilities emerged. Across East Asia, Chlorella was not exotic. It was ordinary. It was something a grandmother might take alongside breakfast without a second thought. This cultural integration did not happen because of aggressive marketing campaigns. It happened because the research was public, the results were consistent, and the populations in question had recent, living memory of food scarcity. When you have experienced real hunger, you tend to pay close attention when credible scientists identify something that grows complete protein faster than anything else on the planet. Then came the space race. And Chlorella's profile shifted from scientifically interesting to strategically critical. NASA's Controlled Ecological Life Support System program, known internally as CELSS, was designed to solve one of the most fundamental problems of long-duration spaceflight. How do you feed a crew for months or years when resupply missions are impossible? Chemical rations had limits. Freeze-dried food degraded. The answer, engineers concluded, had to be biological. You needed a living system that could recycle carbon dioxide into breathable oxygen while simultaneously producing dense, complete nutrition in a confined space. Chlorella fit that description with almost uncanny precision. NASA-funded researchers at universities across the country ran extensive trials through the 1960s and into the 1970s. They measured protein bioavailability. They tested digestibility under controlled conditions. They analyzed vitamin content, mineral profiles, fatty acid composition. They studied how efficiently chlorella converted waste carbon dioxide into usable biomass. The organism performed consistently. It was not a curiosity. It was not a novelty. It was a serious candidate in the most advanced life support engineering program in human history, funded by the same government that would later hesitate to classify it as food. So what happened? Why did an organism validated by Carnegie, Stanford, the Rockefeller Foundation, and NASA end up confined to the supplement aisle instead of integrated into the American food supply? The answer is not contamination. It is not toxicity. It is not that chlorella failed any meaningful test. It is that the system surrounding it had already succeeded at building something else entirely. By the time chlorella research reached maturity, American agriculture had committed fully to a different model. The post-war food economy was constructed on the foundations of corn, soy, wheat, and industrial livestock. Federal subsidies locked in those crops at massive scale. Processing infrastructure was engineered around their specific properties. Supply chains, grain elevators, feedlots, slaughterhouses, and national distribution networks all assumed a commodity grain architecture. Chlorella did not fit that architecture. It could not be harvested with a combine. It could not be stored in a grain silo. It could not be processed in an existing mill or fed to cattle in a feedlot trough. It required photobioreactors or carefully managed open pond systems, which meant new infrastructure, new technical expertise, new capital investment, and critically, no existing subsidy support to offset the risk. The issue was never whether chlorella could produce protein. The issue was whether the existing economic system had any structural incentive to let it. The FDA's regulatory position reinforced this exclusion in ways that compounded over decades. In the United States, the line between a food and a dietary supplement is not purely scientific. It is regulatory, procedural, and deeply historical. Foods that were commonly consumed in the American diet before 1958 are generally recognized as safe under existing statute. Foods introduced after that date face an entirely different burden of proof, requiring extensive petitioning and review. Chlorella, despite its deep global research history, was never a common food in the American household. It was common in Tokyo, it was common in Taipei, but it was not common in Chicago or Dallas or rural Ohio. And so when chlorella entered the American market, it entered as a supplement, not a food. That single classification decision carries enormous consequences. As a supplement, chlorella cannot be marketed with specific nutritional claims the way soy protein or whey protein can. It carries mandatory disclaimers. It occupies a regulatory gray zone that constrains its commercial reach and quietly erodes consumer confidence. The FDA has also issued safety warnings about certain blue-green algae products over the years, and while those warnings technically target cyanobacteria species rather than chlorella, the public does not draw that distinction. In the consumer mind, algae becomes a single undifferentiated category. And that category now carries a caution label that no amount of NASA research seems able to remove. This is a pattern that repeats itself across the American food landscape with quiet regularity. An organism or a crop demonstrates genuine nutritional and agricultural value. Institutional research validates its potential. But the economic infrastructure is already committed to an incumbent. The regulatory framework is built to protect what already exists. And the alternative stays marginal, not because it failed any biological or nutritional test, but because the system was never designed to absorb it. You can trace the same pattern with amaranth, a grain that fed civilizations for thousands of years, and now sits in the specialty aisle. With perennial wheat varieties that could rebuild topsoil but require breeding investments that no subsidy program supports, with heritage legumes that outperform modern monocultures in nutritional density but lack processing infrastructure. The bottleneck is almost never biology. It is almost always economics, regulation, and the invisible weight of decisions made decades ago by people who were solving a different problem. Today, Chlorella occupies an increasingly strange position. The global market for it is expanding steadily. Production technology has improved by orders of magnitude since the 1960s. Modern closed photobioreactor systems can cultivate Chlorella with minimal contamination risk, high batch consistency, and improving cost efficiency year after year. Companies across Europe, Asia, and even parts of the United States are scaling operations. The European Food Safety Authority has evaluated specific Chlorella products and issued favorable safety opinions. Meanwhile, in America, you can purchase Chlorella in capsule form from a health food store shelf, but you cannot buy it as a labeled protein ingredient in a mainstream food product without navigating a dense maze of regulatory barriers that most manufacturers will not attempt. The science has not changed. The biology has not changed. The organism still doubles its biomass every single day. It still produces complete protein with every essential amino acid the human body requires. It still converts carbon dioxide into oxygen with remarkable efficiency. It still demands a fraction of the land and water that conventional protein sources consume. What has not changed is the system built around it. For American households watching grocery prices climb month after month, watching the real cost of animal protein rise with each quarterly earnings report, watching topsoil loss reduce the nutritional density of the same vegetables their parents once relied on, the Chlorella story is not really about algae at all. It is about the distance between what is scientifically possible and what is systemically permitted. It is about who decides what counts as food in a country where that definition is shaped as much by existing infrastructure and historical subsidy as it is by nutrition, safety, or common sense. NASA looked at Chlorella and saw a way to sustain human life in the most hostile environment ever encountered. The FDA looked at the same organism and saw something that did not fit inside the existing framework. Both of those responses are rational within their own institutional logic. But only one of those institutions was asking the right question. And the right question was never whether Chlorella is safe. The right question is whether we can continue to ignore the fastest protein-producing organism on Earth while the economics of conventional food grow less stable with every passing season. That question is no longer theoretical. And the longer it goes unanswered, the more it will cost every household that depends on a system built for a world that no longer exists.